I think. I'm really grateful for the awards committee for selecting two absolutely outstanding speakers. One at the postdoctoral level, Astra Bryant, who's in the lab of Dr. Hallem, and she will represent the postdoctoral students. And then this year we have a new category, since at UCLA, after 5 years of being a postdoc, you're reclassified into a researcher category. We decided to open it up actually to all investigators, researchers who are not in a faculty series. And so in that new category, as an associate project scientist, Jiannis Taxidis will be the inaugural speaker. So today's lecture also honors with that the category of researchers that is usually a little bit overlooked, postdocs and researchers that are on the way of becoming independent faculty, but are not really part of an official program. So I'm really grateful that the BRI, since now a long time, has this series. We're also honoring Arnold Scheibel, after whom this lecture is named. Many of you, as we just discussed, have known Arnie. Many of you have not known Arnie. He was an amazing scientist. He was really interested in the brainstem reticular formation and also the thalamus, looking at both the very detailed neuroanatomy, but also the function, the physiology of these systems. Besides, as the intro slide illustrates, he also was a very inspiring, thoughtful teacher. He had this anatomy class that he was teaching to undergraduates, and the average of the grade was close to a Z, D, C, A, B, C, not a Z, a C. But students really loved him and just thought he was absolutely amazing. And when they came back, they then made up the grades that were in the A and B range. So he was a really inspiring teacher. He also was the third chair of the Brain Research Institute from 87 to 95. And during that time, made some really impactful changes. He started the affinity groups. He started Project Brainstorm. So it was really instrumental in getting the BRI more involved in research and especially also outreach activities. So without further ado, I would like to open the meeting and introduce Astra Bryant. Both Astra and Jiannis have indicated that they would prefer to have questions at the end of their talk. So either type them into chat or speak up at the end of the talk. Chat might be the easiest. If you have a really urgent question that has to do with something you just don't understand, you can also type it into chat and I will try to interrupt them. Astra Bryant has had a really amazing career. She started at Bryn Mawr, where she did, as an undergraduate, where she did research looking at leech and their swimming behavior. And then she went on to Stanford, where she first worked as a researcher in John Hugenar's lab, and then it did a joint thesis between John Hugenar and now I forgot who the other person was, and Eric Knudsen, that's right. So did the joint advice Ph.D. meeting between John and Eric, and then working on, the reticular formation in part, or at least the thalamus. So there is a nice connection there. And then she went on to say, well, now I've worked on leech and then a little bit on mice and birds. Actually, she did some work on chicken. Then she decided, well, Stanford has other animals to offer too, and joined the lab looking at cichlid fish. And actually, out of all of these endeavors, got very nice publications out of it. So after her initial postdoc with Ross Fernald, she then came to UCLA, where she's now working in Elissa Hallem's lab and will tell us all about the neuroscience of yet another invertebrate. So please take it away. Thank you, Astra.
unmuting. Folks can see my screen?
Yes.
Okay. Thank you, Felix, for that very kind introduction. And thank you also to the selection committee for this award. I'm just extremely honored. So today I'm going to be talking about my postdoctoral research in the Hallem lab that seeks to understand the neural mechanisms that underlie temperature-driven host seeking in skin-penetrating parasitic nematodes. So this class of worm is a major source of neglected tropical disease, particularly in the world's most impoverished communities. These intestinal parasites infect approximately 1 billion people worldwide. And examples of skin-penetrating parasitic nematodes include hookworms, as well as the human threadworm Strongyloides stercoralis, which is a major focus of my research. So Strongyloides stercoralis infects around 610 million people. And infections are often asymptomatic, but heavy infection can lead to chronic gastrointestinal distress, stunted growth and cognitive impairment in children, and even death. Current treatments exclusively seek to reduce worm burdens through antihelminthic drugs, but this strategy is often insufficient due to really high reinfection rates. And in addition, there's this ever-growing concern about the rise of drug-resistant parasites. So in the Hallem lab, our hope is that a better understanding of how these parasites find their hosts could lead to new therapeutic strategies for nematode control. Strongyloides stercoralis are infective exclusively during a soil-dwelling, developmentally arrested third larval stage. These IL-3s seek out hosts and infect usually by penetrating the skin of the feet. Once inside a host, IL-3s migrate to the intestine where they develop into parasitic adults. These adults reproduce and their larvae exit the host in feces. Now, back in the environment, the larvae develop into IL-3s and the cycle starts all over again. So other skin-penetrating species, such as hookworms, have a very similar life cycle to this. But among skin-penetrating nematodes, Strongyloides species are unique in that the progeny of parasitic adults can cycle through a limited number of free-living generations that include free-living adults. And these free-living adults are amenable to genetic manipulation using techniques that have previously been developed for the free-living nematode C. elegans. And so this amenability allows us to generate transgenic and knockout IL-3s. And you'll see that I use this in my research to develop an understanding of the neural mechanisms underlying the behavior of infective larvae. So the overall goal of my research is to understand the process by which IL-3s locate and infect hosts. We know that IL-3s can respond to multiple host-emitted sensory cues, including heat and odors. And during my postdoc, I've been working on answering two main questions. What role do thermosensory cues play in driving host-seeking behaviors? And what are the molecular or cellular adaptations that might be underlying any kind of parasite-specific thermosensory behaviors? So today I'm going to show you some data that is speaking to both of these questions. So as the first step to better understand the response of IL-3s to temperature cues, I built a custom heated arena that allows us to track the path of IL-3s migrating in precisely controlled thermal gradients. So key features of this setup include a 22 by 22 centimeter auger surface that the worms can migrate freely on, as well as cameras suspended above the setup that allow us to image the movement of worms at defined time points. Using this setup, we tested the migration patterns of IL-3s exposed to temperatures ranging from below ambient to above human body temperature. And we found that they engage in two distinct behavioral modes: positive thermotaxis, a behavioral mode characterized by attraction to mammalian body temperature, and negative thermotaxis, defined by migration towards temperatures below ambient. So here I'm showing, the lines that I'm showing you here are the tracks of individual IL-3s engaging in either positive thermotaxis, the red lines, or negative thermotaxis, the blue lines. And our current thinking is that worms that are engaging in positive thermotaxis are engaging in host seeking, whereas those that perform negative thermotaxis are engaging in a form of environmental navigation. Okay, so at this point, I'm contractually obligated to show you a really creepy video of the IL-3s engaging in the heat-seeking positive thermotaxis behavior. So what you're going to see are about 1,000 IL-3s crawling in a linear thermal gradient that ranges from 20 to 34 degrees Celsius, which is the approximate temperature of human skin. So I've placed the worms at 30 degrees right here, and each IL-3 is going to look on the video as a single black dot. And as I play it, keep in mind that each IL-3 is 0.5 millimeter long, and the distance from their starting point to the edge of the plate is around 6 centimeters. So as you can see, these worms are moving really quickly towards heat. This is 6 centimeters in under 3 minutes, is pretty good time for a worm that, again, is 0.5 millimeter long. And I just wanted to mention that this is actually the most robust sensory-driven behavior we've ever seen in the Hallem lab in any worm species. Okay, so for the rest of my talk, I'm going to focus on the neural mechanisms underlying this behavior, which I'm going to alternatively refer to either as positive thermotaxis or heat seeking. So to understand the genetic and neural basis that produce heat seeking in infective larvae, I took advantage of the classic nematode model system C. elegans. The molecular and cellular basis of C. elegans thermosensation has been studied extensively by a number of different labs. And Stercoralis and C. elegans share many of the same genes and have broadly conserved sensory neuroanatomy. Despite these similarities, their thermosensory responses are highly divergent. So what do I mean by this? For C. elegans, again, the work of multiple labs have shown that adults don't have a hardwired temperature preference. Instead, they'll navigate up or down thermal gradients to reach a remembered cultivation temperature, as long as that temperature is within their physiological temperature range of 15 to 25 degrees Celsius. C. elegans will also avoid noxious temperatures, which includes anything hotter than around 26 degrees. And these behaviors contrast really strongly with the parasite-specific thermal behaviors that I've observed, particularly the attraction of IL-3s to mammalian body temperature, which is in the 34 to 38 degree range. So the first mechanistic question I asked was whether these divergent behaviors are dependent on similar thermosensory pathways, or whether the parasites have evolved new molecular mechanisms that drive their behavior. In C. elegans, thermosensation within the 15 to 25 degree physiological temperature range is primarily mediated by a cyclic GMP signaling cascade. There are other molecular mechanisms, including TRP channels, that contribute to the avoidance of noxious temperatures. I do want to note here that these pathways are known to function in partially distinct subsets of sensory neurons. So recently, the Hallem lab adapted CRISPR-Cas9 technology for use in Strongylides species. So I used our mutagenesis protocols to disrupt a key element of the cyclic GMP signaling cascade, the Tax4 gene, which I'm pointing to with this arrow right here. So Tax4 encodes a cyclic nucleotide gated channel subunit that in C. elegans is necessary for sensory transduction in general and is specifically required for temperature-driven migration. So what happens when I disrupt the Stercoralis homolog of the Tax4 gene? So when I do this, and I tested Tax4 mutant IL-3s, I found that they showed significantly reduced positive thermotaxis compared to control worms. So here I'm showing you the tracks of individual worms, either the controls on the left or Tax4 mutants in the center. And we can quantify their behavior, quantify the effect of mutating Tax4 by calculating the temperature change experienced by the worms during the assay. I think you can see both in the tracks and the quantification that where the control worms are moving really strongly up the gradient, the Tax4 mutants don't show strong heat seeking. So this result told us that IL-3 heat seeking at least partially reflects an adaptation of molecular pathways for C. elegans thermotaxis navigation within that physiological temperature range. Now, in both Stercoralis and C. elegans, Tax4 is actually expressed in multiple head sensory neurons. So let me orient you to this image, which is showing Tax4 expression in a Stercoralis IL-3. So the nose is to this upper left-hand corner, the tail would be down towards this downwards right corner. And in the fluorescent bits here are individual neurons that are Tax4 positive located in the amphid sensory organ of the worm. And this expression pattern is very similar to the expression that we see in C. elegans. And in C. elegans, the list of Tax4 positive neurons includes several that contribute to thermosensation, including the primary thermosensory neuron pair, which is called AFD. So what I wanted to know was, in IL-3s, does attraction to host body temperature depend on the Stercoralis AFD homologs? Or have the parasites co-opted other sensory neurons? For example, maybe some of these other Tax4 neurons. To answer this question, I first had to solve kind of a historical challenge in the field, which is identifying the Stercoralis AFD neuron pair in the first place. So in C. elegans, the AFD neurons are defined by these finger-like ciliary protrusions, sort of the very tip of their processes. And these processes are thought to be essential for thermosensory function in this neuron. However, EM reconstruction studies from the Schad lab at UPenn found that Strongyloides stercoralis actually lacks any amphid sensory neuron with a finger-like ending. Indeed, there's only a single amphid neuron that has an elaborate process, and it has this lamellar ending, which I'm showing to you in red here. And based on morphology, it's not been clear for almost 20 years whether this lamellar neuron is homologous to the C. elegans AFD neuron or to the AWC neuron, which also contributes to C. elegans thermotaxis. So to solve this issue, I turned to genetics. So in C. elegans, there's 3 receptor type guanylate cyclase genes that are selectively expressed in AFD and contribute to thermosensations through that cyclic GMP signaling cascade. So we identified 3 Strongyloides genes that appear to be homologs of these C. elegans genes. And when we looked at their expression patterns, we found that they're expressed in a single head sensory neuron pair in Stercoralis IL-3s. Furthermore, when I used confocal microscopy to look closely at the ciliary processes, I saw structures that were just stunningly similar to the early EM reconstructions of the lamellar neuron. So this merging of genetics and structure suggested that the lamellar neuron is the homolog of the C. elegans AFD neuron. Okay, so now we can ask, does attraction to host body temperatures depend on the Strongyloides homolog of the AFD neuron? To answer this question, I tested the effect of silencing AFD on heat seeking by IL-3s, which I did by adapting a C. elegans inducible chemogenetic silencing method for use in Strongyloides. So in this technique, I express a histamine-gated chloride channel, HisCl1, under the control of an AFD-specific promoter. And this allows me to inducibly silence AFD neurons by soaking the worms in histamine. And what I saw is that when AFD activity is suppressed in IL-3s, they show a pronounced deficit in their attraction to body temperatures, compared to IL-3s that express the HisCl channel but aren't treated with histamine. These are the control worms. And I'll note that this result is fairly consistent with early laser ablation work of the lamellar neuron from the Schad lab. Okay, so with this, we now have a situation where we have a common sensory neuron, AFD, that contributes to both C. elegans and Stercralis thermotaxis, despite the dramatic differences in their behavior. So are those behavioral differences at all reflected in how the AFD of each species responds to thermal stimuli? For example, we might hypothesize that maybe the Stercralis AFD neurons are specialized for encoding warmer temperatures than C. elegans. To test this and to measure temperature-driven responses in AFD, I designed a thermal stimulator that allows me to perform calcium imaging while exposing an immobilized worm to controlled temperature ramps. And for my stimulus, I am going to use a physiological temperature ramp that actually mimics the change in temperature experienced by IL-3s migrating from ambient to body temperature in our behavioral assays. So using this setup, I collected data such as this. So here I'm showing you the average temperature-driven responses of both C. elegans and Stercralis AFD. So for the aficionados, for both species, I'm using the ratiometric calcium indicator chameleon expressed under the control of an AFD-specific promoter. So what I hope you can see is that in both C. elegans and Stercralis, AFD shows increased activity when I deliver my thermal ramp ranging from below ambient to near human skin temperature. But there are some really exciting differences between the two species. For example, as other labs have shown, C. elegans AFD primarily encodes a temperature threshold. So it's not active below ambient temperatures, and then becomes strongly active in a manner that doesn't clearly differentiate between temperatures above the threshold. In contrast, for Stercralis AFD, we see that neuronal activity continues to increase almost linearly as temperatures increase above threshold. And this suggests that whereas C. elegans AFD is specialized for encoding temperature threshold near ambient, Stercralis could be tuned for encoding a broader range of temperatures, including human body heat. So how might these differences arise? As I mentioned earlier, in C. elegans, there's these three AFD-specific receptor-type guanylate cyclase proteins that contribute to thermotransduction and are thought to act through the cyclic GMP signaling pathway. And also remember that I've told you that in Stercralis, we've found in the genome 3 homologs of these AFD-specific RGCs. And if we do a phylogenetic analysis of the protein sequences, we can see that there isn't a clear one-to-one homology between the C. elegans proteins and the Stercralis proteins. And our thought was that perhaps these sequence differences are reflected in functional changes that contribute to the differences in neuronal activity and ultimately in behavior. So work from Piali Sengupta's lab at Brandeis earlier showed that for C. elegans, 2 of the AFD RGCs are actually capable of conferring thermosensitivity to other C. elegans neurons in a Tax4 dependent manner. So here I'm plotting calcium responses produced by AFD RGCs that have been misexpressed in a C. elegans chemosensory neuron. So the red line here is showing the thermal properties of the C. elegans GCY23 protein. And the blue line is showing you the responses of the C. elegans GCY18 protein. So what I want you to take from this is that between these two genes, each one is conferring sensitivity to a different temperature range. So what about the Stercralis homologs? So for these plots, I'm showing you the average calcium response in the C. elegans chemosensory neuron that are misexpressing each of the Stercralis AFD RGCs. So 1, 2, and 3. Okay, 2 things. First, all 3 of the Stercralis RGCs are conferring thermosensitivity, compared to only 2 in C. elegans. Secondly, whereas in C. elegans only one RGC, GCY18, confers sensitivity to warm temperatures, all of the Sturkrall's AFD RGCs are responding to body heat, sort of in this range up here. So this result suggests that differences in the number and tuning of thermoreceptor proteins may be contributing to the robustness and parasite specificity of their heat-seeking behaviors. Okay, so to conclude, my research demonstrates that IL-3s display 2 parasite-specific modes of temperature-driven behavior, including attraction to human body heat. And these behaviors contrast a lot with the known thermal behaviors of the free-living nematode C. elegans. But despite these differences, we found that parasitic and free-living nematodes rely on the same sensory transduction pathway and inputs from the same sensory neuron pair to drive their divergent behaviors. I also presented calcium imaging results that suggest that Stercoralis thermosensory neurons more strongly encode temperature ranges that span human body heat. And I identified one possible mechanism, one possible mechanism is that there's an expanded number of thermoreceptor proteins in Strongyloides stercoralis that confer the ability, that confer sensitivity to body heat. And this situation contrasts with what we see in C. elegans and suggests that changes in primary sensory neuron responses may be contributing to the ability of parasitic nematodes to seek out hosts using heat. And as a last thing, I just want to note that taken all together, this research is actually the first direct evidence of differential sensory encoding by homologous neurons in parasitic and free-living nematodes. And with that, I want to thank all the members of the Hallem lab, but in particular, the 3 former undergraduates in the lab who directly contributed to this research, Felix, June Hawley, and Jackie Lopez. Thanks also to the funding that's been helping me throughout my postdoc career. And I'm happy to take any questions.
I had a question, if I could ask it really quick.
Please.
So I was kind of curious about the movement of the Stercoralis IL-3s away from, like, you know, the negative thermotaxis aspect, because I noticed in your 2018 paper that yeah, like when you place them at below the temperature that they remember, they move away from the heat and more towards a colder temperature. In the paper, you mentioned something about like a dispersal mechanism, but I was wondering sort of what the understanding of what that property is exactly has sort of changed.
Yeah, so there's, the way I think about it is that there's a tension between the ability of the IL-3s to use heat to find hosts, but there's also lots of sources of non-host environmental temperatures. So the way we think about it is that they also are using, they seem to use positive and negative thermotaxis also to position themselves in an environment where they will be sort of most likely able to respond to like a really strong heat source. So some behavioral things we've seen them do is that if they're below ambient, they will move to cooler temperatures. When they're at cooler temperatures for a prolonged period of time, they show experience-dependent behavioral plasticity, such that worms that are at cooler temperatures become more likely to engage in heat seeking. So one way that we sort of think about it is that in the absence of a really strong source of host heat, so something that's really in the mammalian body range, we think they might be sort of going towards cooler temperatures to really make them, make them more sensitive to host heat, and also to increase the difference between ambient and host body temperature, so they can sort of really have a strong neural response to the heat. Is sort of the way I've been grappling with that.
There's one more question in the chat, and it says, does ambient humidity affect C. elegans chemotaxis and other related behaviors?
Yeah, there is some evidence that, yeah, ambient humidity does alter C. elegans behavior. I can't speak sort of really strongly to specific experiments, but there's known to be sort of effects of changes in humidity on the ability of C. elegans to respond. It's something we sort of think about, particularly in the context of thermosensation for C. elegans. I haven't done any specific studies when it comes to the parasites, though, on the role of humidity.
I have a question. Did you do any behavioral assays with the C. elegans that were expressing the TAX4 from the parasitic worms? Did it change the behavior of C. elegans?
Yeah, we've not done that. We haven't done sort of replacement experiments either with TAX4 or the AFD RGCs. Yeah, it's a great idea. We haven't done it. It would be very interesting to see what that would do.
There's one more question, and that's probably the last question because then we should move on, but maybe Dr. Hallem wants to say a few words too. If she's still online, just unmute yourself, Elissa, and then we can take a few words from you. So do the parasites have a specific preferred temperature below ambient?
Yeah, so Felix, who I mentioned, spent quite a long time looking at this. They don't appear, there's a temperature, what seems to be happening with the parasites is that there's a temperature at which they sort of, the drive to engage in negative thermotaxis starts dropping off. And it's around, let me remember, it's in the range of like 13 degrees Celsius, which is quite cool. That's in the range of, for C. elegans, they avoid noxious cold, and 13 degrees is in that range. They don't seem to have, we haven't found a temperature in C. elegans, when we think of a preferred temperature, we think of a temperature that the worms will go up or down gradients towards. We haven't seen that behavior in the parasites. The nearest they come is there's a temperature at which they will stop moving down, which again is just a very different behavioral mode than C. elegans. But for parasites, that temperature is in the 12 to 13 degrees Celsius range.
Thank you. Elissa?
Yes, I'm here.
Yeah.
You want me to say a few words? Okay.
So I just put her on the spot because I misunderstood how the introductions were going to go. So Elissa was supposed to have introduced Astra, and Peyman Golshani is actually going to introduce Jiannis. So if you just want to say a few words of things I forgot to say.
Okay, sure. Well, first of all, congratulations, Astra. Great talk. So yeah, just to, I guess, add what Felix has said already about Astra. So Astra joined my lab, having been trained as a patch clamp electrophysiologist during her PhD. And from her earlier research, she already had extensive experience recording from the brains of non-traditional model systems, as Felix mentioned, including chickens and cichlid fish. And so in my lab, as you know now, she's been working on another difficult, very difficult and non-traditional system, which is human-parasitic nematodes. And I think her original interest in studying the neurobiology of human parasitic worms stemmed from a desire to apply her neuroscience background to a really understudied topic with very broad public health relevance. So I was thrilled when Astra decided to join the lab. And over the past couple of years, Astra has really pushed this field forward in exciting new directions that are really transforming the field, both from a technical and biological perspective. And so I think this came across in her talk, but really, you know, the fact that we can now monitor neural activity and you know, silence neurons, all of this is due to technical advances that Astra has really driven forward in the lab in collaboration with others in the lab. But it's, she's a major contribution in developing all of these new technologies, which have really enabled us to ask mechanistic studies in parasitic nematodes for the first time. And Astra has also won multiple awards for her research, including the M. John Pickett Award for Outstanding Achievement in Postdoctoral Research and the Best Talk Award at the Southern California Eukaryotic Pathogen Symposium and many others. So again, just congratulations, Astra, on receiving the Scheibel Award.
Okay, congratulations, and thank you for saying a few words, and I will go over to Peyman Golshani.
Thank you so much. It's a really great pleasure to introduce Jiannis Taxidis, who's been a postdoctoral fellow and a project scientist in the lab. Jiannis obtained formal training in physics and applied math, and during his PhD at the University of Nottingham with Marcus Owen and Robert Mason, he did modeling of cortical hippocampal interactions during sharp wave ripples. And he continued to be a computational neuroscientist, mainly working in Christoph Koch's lab as a postdoc, where he modeled sharp wave ripple events and was able to combine his modeling with in vivo data analysis, where he could, based on the local field potential characteristics, he could predict which ripples would be replay events and which not. So that was a really great neuron paper he published before coming to the lab. So interesting story is that Jiannis contacted me, and I knew he was good, so I drove all the way to Pasadena to recruit him. And I think he thought that he would continue to do computational neuroscience in the lab, but suddenly realized that he was going to become an experimental neuroscientist. And not only that, is that he would develop a lot of behaviors and techniques that we'd never ever done before. So he was in for a pretty rude awakening, but he really excelled and was able to develop new behaviors for working memory, and as he will show you, really become a very skilled experimentalist who can not only do experiments, but he built an amazing analysis pipeline and was really able to shape the story that he's going to talk to you about encoding of sensory input and time in the hippocampus during a working memory task. So it's been really great having Jiannis in the lab, and he'll really make a great assistant professor soon. All right, Jiannis.
Thank you so much for this kind introduction, Peyman. And thank you, Felix, and to the committee for giving me the honor to be the inaugural Scheibel lecturer for project scientists and research assistants, especially on a crazy historic day like today. Let me share my screen. Can everybody see this? Okay, let me start my timer. So let's try to forget the election for the next 30 minutes and focus on remembering. The overarching question of my talk is basically this question: how does the brain remember what happened to us and when? At the end of the day, we can all sit down and pretty much remember all the basic events that took place that day, and we can also put them in more or less the correct order and sort of gauge what the time between these events were. For example, even though I have a newborn that's just 1 month old, which you may hear in the background crying at any point, I can sort of recollect what I did yesterday. For example, I woke up from the baby screaming like every other morning, and then I had to change the baby, and 30 minutes later I fed the baby, then I made breakfast. I know I worked for a couple of hours, and then the baby needed to be changed again, and then me and my wife had to feed him. And from then on, it all becomes a blur of changing the baby and feeding and changing and feeding and changing and feeding. And then you enter a full-blown Groundhog Day mode. But I can still recollect the major events of yesterday. How does the brain do that? Well, we can start by saying that we know at this point that the hippocampus is necessary for this. We know that because For example, if you're asked to, let's say, walk around the university campus and somebody asks you to do a series of tasks. So as soon as you get here, they're like, okay, pick up this coin from the floor, here, drink water from this fountain, buy this item from this cafeteria, and so on. Once you're back at the base, if they ask you, what do you remember and in what order, you can probably remember most of the things that you did, and you can probably report them in the correct order, so that if this on the x-axis is the actual time these events took place versus what you report on the y-axis here, the black dots is probably what you're going to do. You're going to pretty much correlate them very nicely. If, on the other hand, patients with hippocampal damage, when they did the same thing, not only they had problems remembering a lot of these tasks, as you might expect, but they also couldn't place the things that they did remember in the correct order. So the time that the events took place versus the time that they reported in white here was completely uncorrelated. So instead of saying, how does the brain remember what happened to us and when, let's restate the question as, how does the hippocampus remember what happened to us and when? Again, to make the question a little bit more targeted, we can restate it as, how does the hippocampus form a model, a representation of the sequence of our experiences? So you have, an event takes place, and then some time interval, there is another event that we want to remember or we need to remember. There is another temporal interval, another event, and so on. How does the hippocampus form a representation or a model of this sequence of events? Let's simplify it a little more. When you have an experimental animal like a mouse, you can turn this question into, let's say there was a sequence of specific stimuli, an odor stimulus followed a little bit later by a visual stimulus, let's say, and then another odor stimulus and so on. How does the hippocampus form a representation of these specific sensory cues and the time in between them? Well, we know that the hippocampus can form representations that come more or less in two general flavors. One is sensory representations, encoding cells encoding specific sensory inputs coming in, the external world, if you want. So for example, if a mouse has to remember a sound followed by an odor in order to perform a task, the hippocampus will form a sequence of cells that will represent this specific sound cue. And then when the odor comes on, another sequence of cells will start that represent that specific odor. These are cells that encode a very specific, robust sensory input coming into the brain from the outside world. For the time intervals in between, we know that the hippocampus generates also representations of the second flavor, let's say. Let's call them internal representations, because these are not triggered by a very specific sensory input. They're internally generated. And these can be the place cells, representations of place that people are very familiar with, but there's also time cells. For example, if a rat has to remember a specific cue and gets trapped in this part of the maze here for a few seconds before it responds to the cue, then as the rat is trapped here, the hippocampus generates this sequence of neurons. These rasters here show the activity of each neuron across trials. It generates a sequence of these neurons that fire one after the other. Now the rat is not moving around, nothing is happening. So the only thing that is passing is time. So we call these time cells because we, the decoder or a downstream brain region, can decode time based on this activity. How much time has it been since I got locked in here? So you have these two kinds of representations of sensory cues and temporal intervals, time. And you could say that, okay, so if I have cells that encode for this odor, cells that encode for this visual cue, and time cells in between, then by looking at this sequence, I can tell what happened, right? If I'm a neuron or a brain region reading this activity, and I know exactly what each neuron represents, then I can say that, oh, at this point, an odor came on, and then there was like I don't know, 5 seconds, and then I can see that a visual cue came on, and then there was like 10 seconds after that visual cue, and so on. And you can later recreate that sequence of experiences just by reading this sequence. That led two of like the main big thinkers in hippocampal research, Gyuri Buzaki and the late Howard Eichenbaum, to postulate that these hippocampal sequences form trajectories in memory space, tracking sequential experiences, because again, a mechanism that can read this sequence can sort of recollect what the sequence of experiences was earlier. Okay, so now that we have this framework, there is a major question. that is unanswered. And the question is, are the external and internal representations governed by the same dynamics? In other words, does the hippocampus use the same rules, the same modality, the same stability dynamics and so on to encode these specific sensory cues versus the more like fluid temporal intervals in between? And this is an unanswered question because most people study either these sensory representations in the hippocampus or place representations or time representations, but rarely together. So I wanted to ask that question when I joined the lab. And basically to break this question down, I split it into like a series of, let's say, simpler questions. So the first question is, first of all, can sequences indeed combine representations of external cues and time intervals? So to do that, I needed a task that includes both specific, most interesting memory-related cues and temporal intervals. So we trained mice, head-fixed mice, to perform a very simple olfactory delayed non-match to sample task. At the beginning of its trial, we give an odor to the mouse. It can be either odor A or B. The mouse needs to remember that odor for a 5-second delay period where nothing happens. And then another odor comes on. It can be, again, either A or B. If the two odors do not match, so it's an A-B or a B-A trial, then there is an available water reward that the mouse needs to lick port to release. If the two odors matched, there's no reward, and the mouse needs to learn to reject and not lick the lick port in those trials. Now, the actual question, the money question is, how stable are these representations that I said, the sensory and temporal representations across days, or when something in this temporal context of this trial changes? And to address the question of stability, especially across days, you need to track the activity of neurons for multiple days over and over again. So we have head-fixed mice on a spherical treadmill that they're free to run whenever they want, and we perform 2-photon calcium imaging on them that allows us to track the activity of neurons for multiple days. So what I do is I remove the cortex overlying the dorsal hippocampus. I expose the CA1 pyramidal layer. I put a cover slip in a titanium ring. The mouse performs this task under the 2-photon microscope. So if I'm lucky and I do a good surgery, I can expose thousands and thousands of neurons in the CA1 pyramidal layer. These are transgenic mice expressing TD tomato in all their interneurons, shown here in red. So I can then remove the interneurons from my analysis, and everything I'm going to show you today is on the pyramidal cells, shown in green. So just an example of a trial and our setup. This is an A-B trial. So the mouse is free to run, receives odor A in yellow. At the same time, we're imaging here. And it needs to remember the odor, and then odor B comes on and licks to get a reward. If, on the other hand, you have a B trial, so a match trial, there's no reward. The mouse receives the first odor, has to remember it, and later will reject. So as this is happening, we're recording videos like this where we pick up the calcium transients of these thousands of cells as this is happening. And now the last question is, do these sequences emerge during learning of the temporal context, or do they pre-exist? In other words, are these sequences sort of already existing in the hippocampus and they're getting assigned to a trial or an odor and so on, or are they formed together with as the hippocampus learns this task? And to do that, you need a task that doesn't take months and months for a mouse to learn, because it would be unfeasible to image these neurons for that long. So we need a task that can be learned relatively fast. And lucky for us, this task can be learned within 5 or 6 days, by which time mice become experts in the task and do like 90 to 100% correct trials. Okay, together with my friend Eftichios Pneumatikakis at Simons Foundation, we developed a numerical toolbox that became part of the Peyman open source toolbox for motion correcting these videos, segmenting, extracting the activity of pyramidal cells, shown here in blue, removing all the interneurons from our analysis, shown in red. We extract the calcium traces from the pyramidal cells, which we turn into a proxy of firing rate, which I will be using from here on. So what do we see? Let's split the trials into the ones that start with odor A and the ones that start with odor B. So it doesn't matter what the second odor is, it can be either one. Now, what we found was that during these odor A trials, there's one starting with odor A in yellow here, we get a sequence of neurons that are tiling the entire trial from the odor onset until the next odor comes on. So you have a bunch of cells here, and each row here is the average firing rate of a neuron during all these trials. So you have all these cells here that are firing right at the odor presentation, encoding the actual stimulus, and they're followed by a sequence of neurons firing one after the other, tiling the entire delay. So if you were to look at a neuron that has an average firing rate like this one, and you looked at each trial, you would see here, I've stacked the trials based on the combination of odors. This block here is all the AA trials. So you can see that this neuron likes to fire when odor A is presented. It doesn't care if it's the first or the second one in the trial. It just fires right at the odor A presentation, not when odor B is presented. If you look at a cell here, a time cell like this one, it'll fire about 2 seconds into the delay, but only after odor A is presented, where it's going to be more or less quiet if odor B is presented. And that's why this sequence doesn't show up when you look at the same neurons, but for the opposite trials. For those cases, for those trials, you have another sequence composed of its own odor and time cells. So a cell here will fire when odor B is presented. And a late time cell out here is going to fire towards the end of the delay, more or less, with a significant field, but only after odor B, not in trials that were initiated with odor A. So you have two groups of neurons living sort of in a salt and pepper distribution in the pyramidal layer in C1, one forming a sequence for initiated by odor A, the other one forming a sequence initiated by odor B. And you can easily sort of guess that if you train a Bayesian decoder or a support vector machine, a decoder to decode odor or what the odor was or what time it is into the trial, if you use these activities, you can easily train a decoder, and you can always decode time or odor from the collective activity of these cells. Okay, so that kind of answers the first question. Can sequences combine representations of external cues and time intervals? Yes. I've shown you cue-specific, odor-specific sequences representing odor and time. How stable are they across days or changes to a temporal context? So let's start with a temporal context change. The easiest thing you can do is to change the sort of structure of the trial is to extend the delay from 5 seconds to 10 seconds, which is what we did. So if you have an initial sequence, this is the first odor in the trial, the second odor, and these are the fields, the specific fields of the neurons composing the sequence. Then if you extend that delay here from 5 to 10 seconds, various scenarios could take place. One is that the odor cells will generate the sequence just like before. These time cells essentially encode how much time has passed since the odor was given, and the sequence doesn't really care that you added an extra 5 seconds at the end here. So you'll have a replay of the same sequence. Another possible scenario is that these time cell sequences are shaped by the actual interval of that delay, the exact 5 seconds, and they encode that interval. And if you extend that interval to 10 seconds, the sequence will spread out and cover this new interval here shown in. So from the original black sort of configuration, you're going to go into the yellow one. And the last case is that these sequences are formed by the actual temporal context of this delay. And if you change the delay to 10 seconds, you create a different temporal context. And then this sequence will sort of disappear. Most cells will lose their field. They will not be encoding any specific time point. They'll just randomly spike. Or they will switch their field forward or backward in time, and the original sequence will be completely scrambled. So let's see what happened. This is the firing rates across all trials from 3 example cells. And I'm only showing you here the trials that have 5 seconds, the default delay. This is an odor cell firing at the odor presentation. The dashed line here represents the significant field, the point of the significant field. This is an early time cell firing just at the onset of the delay. This is a late time cell firing with a significant field at the end of the delay. As soon as we extend the delay from 5 to 10 seconds, we see this. The odor cell retains its field. It retains a statistically significant field and fires at the odor presentation very nicely, just as before. The early time cell becomes almost completely silent. The late time cell, after a few trials, becomes completely disorganized and fires randomly without a significant field anymore. So if you look at the whole sequence, You have a nice default sequence for your default 5-second delays. And if you look at the exact same cells, stacked the exact same way for the 10-second trials, you see that the odor cells are still there, significantly encoding the odor, whereas the sequence is completely gone. Most cells have lost their field or have completely remapped. Now, these trials have their own sequence, which looks like this, composed of mostly new cells that initially were, they were not encoding any specific time points. So these cells were sort of lurking in the background and immediately jumped in and formed a new sequence. You can see an example here of a early time cell for the 10-second delay trials that was more or less quiet before, and here is another one. So these cells jump in and form a sequence to encode for the new temporal context of the 10-second delay. So what I've shown you is that you have very stable order representations and very flexible time representations that sort of argue that these sequences encode a specific temporal context. And you could say this is maybe this remapping is because you just extended the duration of the trial, not just the delay of the entire trial, and these sequences get messed up. Actually, we did another experiment, which I'm not going to show you here to save time, where we extended just the order presentation and shrunk the delay so that the whole entire trial stayed exactly the same in duration. But we see exactly the same thing. The odor cells remain very stable. They don't care that we extended the odor presentation. Time cells get completely remapped. Okay, what about across days? How stable are these sequences across days? To answer that, we follow the exact same field of view for multiple days, and we match the cells between each pair of days. So we find the cells that were active on, let's say, day 1 and day 6, and we find which of them are active in both cases, and we match them. And what we find is that on any day, you can have a nice sequence like this one with your odor cells and time cells. And if you look at these same neurons, again, stacked the exact same way the next day or the day after, the odor cells more or less retain a significant field during the odor presentation. They're still there, whereas the sequence of time cells is completely messed up or partly messed up the next day. It has remapped, and by 2 days later, it's almost gone completely. So again, stable odor representations and very flexible dynamic time representations across changes in the context or across days. Now, finally, how do these sequences emerge during learning of a temporal context? To answer that, we image neurons as the mice were learning these tasks. Like I said, initially, we're lucky because the mice can learn this very quickly within a few days. So here is the performance of 11 mice as they were learning this task. On day 1 versus day 6, they went from about 60-70% to 100% performance. Now, if I image these mice on day 1, when they're first learning the task, I get a sequence like before. The dashed line here separates the odor cells firing during the odor presentation and the time cells firing during the delay. If I image the exact same mice, exact same field of view, same neurons, 6 days later, when the mice are experts in the task, I see more or less the same number of odor cells as before, about 150 in this case, and way, way more time cells. In fact, if I just count how many odor cells do I get every day versus how many time cells do I get every day, I see that the number of odor cells remains pretty much flat, whereas the number of time cells increases as the mice are getting better and better. So if you just do a correlation between the number of odor cells and performance, there's nothing, whereas the number of time cells are very nicely correlated with the performance. As the performance improves, more and more time cells are emerging. Now, it could be the case that learning this task has nothing to do with this. It could be that because we're giving mice these odors over and over and over again for 6 days, we're triggering the odor cells, and just the passive plasticity mechanism creates more and more downstream neurons that form longer and longer sequences, right? To address this possibility, we repeated this experiment. We gave the exact same trials for the exact same number of days to a different group of mice that were completely untrained to this task. They were not even pre-shaped, let's say, to know that there is a water reward, there is a lick port there, nothing. They were receiving the same odors, but they just didn't know what to do with them. So they were just passively sitting there smelling the odors, right? So again, we're triggering the same odor cells just like before in theory. And if there is a passive plasticity mechanism is at play, then these sequences will just expand. But we did not see that. On day 1, we find, some odor cells and time cells in these mice. And on day 6, we find pretty much the same number of odor cells and the same number of time cells, so that now both odor cells and time cells their numbers remain flat. They don't change across days. So that these naive mice end up with significantly less time cells than the mice that were learning the task. A final question that I wanted to address is, okay, so I showed you that time cells increase as mice are learning the task. I also said before that every day we get a practically new group of time cells, right? So put these pieces of information together, it means that every day as the mice are learning, a relatively new group of neurons comes on, becomes time cells, and it's getting bigger and bigger. So the question is, all these new time cells, what were they doing before? Were they just silent? Were they randomly spiking? What happened? How do they emerge? So let's track the activity of some of these cells across all trials, all days, right? This is a cell here that becomes an odor cell on day 2. This is what the white dashed line indicates a significant field at that point, whereas the blue dashed line shows you where the peak firing rate is across that day, but it's non-statistically significant. So that guy spiked for a few trials in the first day, non-significantly peaking here, whereas on day 2 it already created a significant field that it retained for the next 5 days. So a stable odor cell. This is a cell that was a significant time cell on day 5, shown here by the white dashed line. And if you go back in time, you can see that the firing rate sort of dissolves, becomes more and more noisy and sparse, so that the peak firing rate sort of moves away from it. And even the next day after it had that field, it started moving away and lost the field. So if you look at some other examples, this neuron, for example, started firing on day 2 and then moved its peak towards the end of the delay and started firing more and more. And then by day 5, it has a nice, significant, strong field there. And then the next day, it's starting to move away again. Same thing, I don't know, for this cell here, fires towards the end on day 1, and by day 2, starts moving towards the beginning and forming a field, but day 3, it's a significant field, and then starts drifting away. So the key point here is that most of the odor cells look like this, whereas most of the time cells look like that. In other words, the odor cells very quickly attain their field, so that they don't, this peak activity doesn't travel around too much. It just falls into the odor field and stays there. Whereas most time fields sort of have this drifting peak of activity and form these long trajectories, so that the time cells have significantly longer trajectories than odor cells. But the other interesting thing is that each day, these cells do not fire randomly at the beginning or the end of the trial and then just shift back and forth completely randomly. They sort of form these more smooth trajectories, sort of slowly getting into their field and slowly diverting away from it. That means that you have a slow drift in the population, so that if you take the sequence on any day, in this case, I'm taking the sequence that I had on day 5, the odor cells and the time cells on day 5, and then I look at the activity of the same neurons going backwards in time on day 4, 3, 2, 1, or the next day. Every day I'm replotting the template here just for reference. And in blue, I'm showing you the peak activity of these neurons, but that it's not significant. And yellow means that that cell shifted its activity to that new point, but it's a statistically significant field. And what you can see here basically is that as you move backwards away from this configuration here, less and less yellow, so less and less significant fields, and the sequence seems to be devolving away from it. And the same thing happens if you move to the next day. So that day 4, you can sort of see an echo of the sequence. If you go further back, it's pretty much gone. You can easily quantify this by saying how far am I firing versus the field that I'm going to have on day 5. And you can see that, as you go backwards in time, the cells monotonically move away, become more and more distant from their field on day 5. And the same thing happens the next day. The same if you just use the sequence of day 3, cells would slowly drift into their configuration on day 3 and move away. If you look at day 1, your initial sequence sort of dissolves across days, Whereas if you use the cells that are sequenced on day 6, you see that they slowly just monotonically get into their configuration on day 6, and then on day 7 they would completely start devolving again. You can use Bayesian decoding and you would get the same thing, so that information forms and then deforms across days. So that sort of answers the last question: do time representations emerge during learning? And I've shown you that yes, they emerge during learning of a temporal context, and they're in a constant evolution, a constant population drift. This can be summarized by a graphic, which I'm going to skip in the interest of time. So I started this by asking, are external and internal representations, sensory and time representations, governed by the same dynamics? And I hope I've convinced you that the answer is no. I've shown you very stable representations for sensory, external cues, and very flexible, dynamic, learning-dependent representations for the variable temporal intervals in between these cues. And I would argue that this is an optimal strategy. It is a necessary combination. The stability and flexibility is a necessary combination for the hippocampus to construct these maps of sequential experiences in memory, so that, you know, every time I change my baby or I feed the baby, the hippocampus would arguably use the same cells to encode the baby cells, whereas it will be able to use a different sequence and generate different representations easily and readily for those very short and very, very precious short intervals in between these tasks. And with that, I would like to thank pretty much everybody in the lab that has helped in one way or another, current and previous members. Of course, Peyman for his mentorship, my friend Efthymios for helping with the calcium, and a long list of excellent undergrads that have helped with training these animals and even running experiments. This work was published in Neuron a month ago. And thank you so much for your attention. I'm happy to take questions.
So very interesting talk. I'm just wondering, do you know the answer to this question if not only the neurons themselves are dynamically regulated during your assay, but if those neuronal cell types in those neurons that are activated are also dynamically changing?
Neuronal cell types, you mean pyramidal cells versus interneurons? What do you mean neuronal types?
Yeah. Sorry, I wasn't clear. So the chemical phenotypes inside that reside in those neurons, do you think they're also dynamically calibrated in the same manner or different manner? So I guess the chemicals that reside within the neurons, like the phenotype.
I think I see where you're getting at. So basically everything I showed you again is pyramidal cells only, and I would be very interested to see what happens with interneurons. And that's like one of the main classifications, right? And then the interneurons are also split into a long list of different cell types with different circuitry that I'm very curious to sort of dissect their role into shaping and controlling the evolution and emergence of these sequences. But beyond that, I wouldn't be sure, because we didn't address that, the chemical sort of phenotype of these cells. But I wouldn't be surprised if beyond sort of like circuit mechanisms, there is also sort of neuromodulatory mechanisms that sort of control which cells will acquire a field and which not, which cells will fire during an odor and which not, and so on. But the circuit or the neuromodulation that or excitability that controls that is still unknown.
Great talk, Jiannis. I just had a quick question. So you presented them with the 10 seconds delay, and I was just wondering if you presented them with the same delay over a long period of time, do the time cells come back or do they get ordered? And say if you are following the 10 seconds, if you present it with the 5 seconds, does it remember the 5-second delay at some point?
We did do that, but I didn't do it very systematically, so I can't really say. You're asking if I switch back to the 5 seconds, if the original sequence would re-emerge, right?
Yeah, exactly.
I haven't really looked at that yet. We have some recordings like that, but I didn't do a lot, and it's really hard to assess this in with just a few trials. You really need because there is sort of variability in the activation of these cells. These time cells don't show up in every trial. You need a lot of trials to be able to assess that. And I don't think I have enough to safely say what happens. But I would argue, I think what I saw anecdotally, I should say that I think, yes, they do reemerge the original sequence, but don't hold me accountable on that. I'll have to look again at my data.
Got it. Thank you.
There's a question, what percentage of all the observed pyramidal cells are odor or place cells?
In each sequence, I have about 10%. So overall, about 20% of pyramidal cells become a member of either one of these sequences. Now, again, this depends on and these are cells that are statistically significantly have an odor field or a time field and so on. So depending on how you define that statistical significance and what criteria you use, you can be a little bit more lax or more strict, and you can allow more cells to be sort of accepted as having a significant field, or you can be super strict. But using my criteria, which are pretty much usual criteria, benchmark standard type, you get about 20% of the cells. The other 80% of neurons either don't meet this criteria, they fire randomly, and then you also have a bunch of cells that are completely quiet during this whole time, that are not even picked up by the code, because our code only picks up the cells that flash, so that are active.
So then to add to that, I think sort of the classification between when you drew your 150 cells, between those are odor cells and those are time cells, that cutoff seemed to be a little, arbitrary. So do you have ever an odor cell that becomes a time cell, or a time cell that becomes an odor cell?
Yeah, you do. So you have not that many, not surprisingly not many, in the sense that it's not just a random like flow in between those two classes of cells, where a cell can randomly just switch from being an odor cell to a time cell, or vice versa. You do have some, and if I remember,
so to just continue to ask there, if you had, if you did a odor versus taste versus time, would you expect that there's more intermixing of odor and taste cells, and that time cells are sort of their own category?
No, I would.
Okay.
I would expect that if you do something as radical as introduce a whole new sensory cue, like a taste, then you would probably get new cells that will encode that taste. I would say, as long as it's important to the task. I don't know what happens if it's completely unrelated to the task, because I haven't done that. If it's something that the hippocampus is interested in, it will assign cells to it. It will assign cells that are the taste cells. And it might reconfigure the time cells in between, the odor and the taste and so on, but it doesn't, I don't know. I don't know if that answers your question really. If I'm not sure I understood.
Sort of thought, is there some categorical cell that cares about color, taste,
I see.
blood pressure, about kind of those versus more fluid cells that might care about time or sequence or things like that?
I would argue that no, I don't think the hippocampus has preordained cells that are, sort of used as backup cells for whenever that odor shows up, and it has those cells, predetermined in the circuit, and will use them for whenever that odor shows up. That's more like olfactory bulb type of activity. The hippocampus will basically, and that's not something that I've studied, but it's sort of other people have studied before me. The hippocampus will basically generate these representations in order to solve the task. These representations are there when these odors are important to the hippocampus. They have a memory sort of weight. These time cells will be generated when those temporal intervals are important. Like I showed you, the more important the temporal interval becomes as the mouse learns, the more cells the hippocampus throws at them. But these representations are very fluid in that sense. So I wouldn't be surprised if you take the same mouse and you put it in a maze, and you ask it to do a spatial task with visual cues and so on, and these same cells are suddenly assigned to encoding something different. But that's also something on the to-do list in my future directions.
One last question from Astra. Do you see differences in the time cell representation on correct versus incorrect trials?
Excellent question. We actually don't. Other labs before that have looked at these time cells have managed to get some sort of correlation between these sequences and whether the trial is going to be correct or not in different type of tasks. In my case, we don't. I don't think I have the slide here, but basically I've done extensive analysis trying to compare correct and error trials, trying to decode whether I can predict if it's going to be a correct trial or not, if the time representation is more like messed up in an error trial, if the odor was not encoded as well in an error trial. No, I don't find anything interesting in that sense, so that these sequences do not correlate with the correct working memory activation. What do they do, and what is the relation with memory? I would argue that it's more along the lines that I mentioned here, that they're not used in order to solve a working memory task like this. They're used to create a sort of map of what happened before.
So thank you both. Thank you, Astra and Jiannis, for absolutely amazing, wonderful distraction from the other things that are happening today. It's really fun to think about science and not what's going on in the world. And hopefully at some point, science will also influence what's going on in the world. So thank you both and congratulations.
Thank you.